Good morning, everybody. Welcome to the MPG Primer. And today I have the pleasure of introducing Masahiro Kanai. He is a research fellow in Dr. Remnick Xavier's laboratory at MGH and at the Broad here. He previously completed his PhD in biomedical informatics at Harvard Medical School, where he was co-advised by Doctors Mark Daly and Hilary Finucane. As a statistical geneticist, his research focuses on identifying causal genetic variants underlying complex diseases and traits across diverse population. Additionally, he leverages multi-omics approaches to functionally characterize these causal variants and aims to elucidate the molecular mechanisms driving complex diseases. If you have questions throughout, please come down. The microphones are here on the side again. And yeah, take it away, Masa. Thank you.
All right, thank you so much. And thank you so much for joining my MPG primer this morning. I'd like to talk about fine mapping complex traits in biobanks. So for the first part of my talk, I'd like to give a brief introduction to fine mapping. Before diving into the details of the introduction, I'd like to give a special thanks to my colleague, Ran, who have been working together with me for many fine mapping projects. And she did a great MPG primer 2 years ago and generously shared a couple of her background slides for my talk today. So as many of you are familiar with, GWAS have been very, very successful at finding associations in the last 20 years. This is the latest schizophrenia GWAS from the PGC consortium. There are around 300 loci that are generalized significant. But if you really zoom into each of these significant loci, you may realize pinpointing a causal variant is really challenging. This is primarily because of these genetic variants are correlated to each other, which is called linkage disequilibrium or LD. And this correlation makes it harder to identify which one of these correlated variants are actually causally associated with a disease. So many of the fine mapping methods aim to nominate causal variants based on this scenario. Now you have a GWAS data, which is observed, and there should be a causal SNPs, which is unobserved. What we observe is basically the representation of these two causal variants. And due to LD and noise, we observe this kind of a locus plot. And fine mapping methods basically disentangle this LD structure into independent signals, aiming to identify each of these causal variants. So suppose this is a goal. Many fine mapping methods output two things. First output is called posterior inclusion probability, or PIP, which is the probability that a variant is causal given the observed data. Second output is a credible set, which means to capture the uncertainty of the identity of the causal variants due to LD. So you often see a credible set, 95% credible set or 99% credible set, which basically means these credible sets capture 95% of the PIP for given independent signal. And fine mapping methods basically output these two outputs, and we heavily rely on these outputs to interpret which causal variants or which signal is more likely to causal. So based on these concepts, now I'd like to give a brief overview of the fine mapping methods. I think there are two major axes to categorize these fine mapping methods. The first axis on the y-axis is basically whether you model single causal variant or multiple causal variants. And the second axis you can see on the x-axis is basically whether a method is primarily for a single cohort or multiple cohorts. And for multiple cohorts, there's a distinction whether your analysis is based on the meta-analysis or joint analysis or mega-analysis. I don't dive into details into each of the methods, but I would like to cover some of the major methods here and give you a basic concepts behind it. So the first and the most straightforward one is this method called ABF. ABF, or approximate Bayes factor, is basically a Bayesian method based on Bayes factor for each variant. This is pretty simple, as you can see in the equation here. This method basically assumes a single causal variant in a locus. So in most cases, these Bayes factors are roughly a monotonic transformation of the G-scores that you observe in the GWAS, and highest PAP are almost always given to the most significant variant. This is, very simple method, but still powerful or robust method that you can rely on in many cases, especially if you don't have access to the LD matrix or good external LD difference panel. ABF method is the first method you must rely on for your summary statistics. But of course, because we assume a single causal variant in a locus, this is a big limitation of the ABF method. So suppose in a scenario like you have 3 variants in this particular locus.
Great, there's one tray.
2 variants called purple and red are causal variants. Gray variants in the middle is just a non-causal typing variants. But if these 2 causal variants are weakly tagging the middle non-causal variants, and the 2 causal variants are independent of each other, then ABF assigns the highest PAP to the non-causal variant because this non-causal variant is tagged weakly by 2 causal variants and having the highest G-score. Similarly, in a scenario where you have 2 causal variants in a locus, but neither of these 2 causal variants are genome-wide significant because these 2 causal variants are correlated to each other and have opposite true causal effects, or they have same causal effects that correlation negative between the two. And in this case, basically LD cancels out these causal effects, and ABF assigns a low PAP to the non-significant associations. And this might sound like a hypothetical situations, but we do actually observe such examples in the real data. So here, this is in the chromosome 5 in FGFR4 locus. For height, we find mapped in 3 biobanks, BBJ, Finch, and the UK biobank. If you focus on this ROS 244711 variant, this variant is actually fine mapped in every cohort, but not genome-wide significant only in Biobank Japan. If you look closely into the underlying causal structure, we basically found out that there is another missense variant of the FGFR4, which is also fine mapped in every cohort. And this missense variant has a negative correlation to this RS244, the second causal variant, only in the Biobank Japan. So only in the Biobank Japan, the causal effects are canceled out, and the secondary causal variant is not genome-wide significant in the BBJ. So this clearly illustrates we have to model multiple causal variants for whatever fine mapping method you use. for highly powered rate. So this is our natural introduction to the second class of the fine mapping method, which I'd like to cover today, which is basically the single cohort multi-causal variance fine mapping method. There's plenty of the multi-causal variance fine mapping method for a single cohort, but if you ask us to characterize into three categories, I think the first category is basically a conditional analysis. This method is sometimes called causal ABF. Basically, causal is a method that you can do a conditional analysis on the summary statistical level. And ABF is a method that I described earlier. So suppose you observed this kind of a logism plot from your viewers. If you condition the lead signal using causal, then you might observe the secondary signal independent from the first lead variant. Causal ABF simply applies ABF method to the first summary statistics that conditioned on the second variant, and a second summary statistics that conditioned on the first variant. So that by doing a conditional analysis, you can get the approximate independent signals for each of the method. This is a kind of a powerful approach that has a one big caveat. This method basically doesn't handle the situation like the one left, where your non-causal variant is tagged weakly by multiple causal variant. In this case, the lead variant is a non-causal variant, and if you condition on the non-causal variant, then you basically lose independent signal for both of the causal variants that are weakly tagged. So there is a more sophisticated method which directly model causal configurations, such as Caviar, Caviar-BL, FineMap, and DAPG. So suppose you have M variants in the locus. There are 2 to M combinations of whether each of the variants in the locus are causal. You typically limit how many causal variants or how many ones in this gamma array can exist together. And you're going to compute this probability based on some algorithm. Most of the prior methods before FineMap doesn't scale well, but FineMap proposes a very computationally efficient algorithm called shotgun stochastic search, which basically enables Biobank-scale fine mapping.
And the pastries don't have to be.
Yep.
This is on your screen.
Close this.
That's hiding some of the. Thank you.
Sure. It's fine. All right. Where is this? Okay, it's the third category of the multiple causal variant fine mapping method is some of the single effects model or SUGI. Basically, in this model, instead of creating a single gamma causal configuration, you divide your causal configuration into L independent single effect causal configurations. And they basically develop empirical-based iterative. They use their iterative Bayesian stepwise selection or IBSS method to compute a positive distribution of these single effects. And this substantially speed up the computation compared to their previous methods. And basically, these computationally efficient methods, such as FineMap and SUGI, both are published in 2016 and 2020, have finally enabled Biobank-scale file mapping, like in the scale of the 500,000, like a UK biobank.
Copy.
And this opens a new era of the file mapping, in my opinion. Just file map every trait in each biobank. So I'd like to switch my gear and talk about the file mapping in the biobanks. There are a couple of motivations why we want to file map in the biobanks instead of a single, disease consortium or just a single, a prospective cohort for one trait. First obvious reasons is now in the era of the biobanks, we have substantially higher, bigger numbers of samples and substantially bigger numbers of phenotypes that are systematically collected in each biobank. These are just examples, but you all familiar with the UK Biobank, FinGen, Autobus, Medium Veteran Program, Biobank Japan. All of them have more than hundreds of thousands of the samples and hundreds of the phenotypes that are systematically collected within each biobank. And this enables us a systematic comparison of the file mapping and the causal variants across the diverse populations. Actually, in a global effort called the Global Biobank Meta-Analysis Initiative, we are able to collaborate with 23 biobanks around the globe, totaling 2.2 million individuals. This is data from the 2022, and I believe now we have around 30 biobanks, more than, well, I don't remember actual exact numbers, but these numbers are growing. And this made us, this empowers us to do a file mapping at the scale. The second motivation is we do expect more file mapping discovery by starting more diverse cohorts. So in human populations, low frequency variants often tend to be populations enriched or sparsic. And some of these variants are even under selection. So this is one where my favorite example, alcohol thrush and the ALDH2 variant called RS671. If you drink alcohol, or ethanol to be specific, there are two genes that are basically involved in metabolization of alcohol. If you have this RS671 variant, basically that inactivates ALDH2, and these two are basically a phenomenon called alcohol thrush. This particular variant is selected in East Asian populations. In the East regions, 26% minor frequency. In the rest of the world, less than 0.1%. And moreover, because drinking behavior is heavily associated with your diet, this RS671 associated with esophageal cancer and many other diseases, including your food dietary pattern, like whether you are more likely to eat cheese, drink a milk, or even a natto, which is fermented soybeans. Third motivation is we're gonna expect a higher file mapping resolution. As you know, different ancestries have a different LD length and regions. So suppose we have two populations that have a slightly different LD length and LD regions. By combining multiple populations, you're gonna expect to narrow down basically an LD region and narrow down the causal variance. This will highly improve the file mapping resolution. So based on these motivations, there are multiple methods have been proposed to conduct a multiple cohort fine mapping or multiple population fine mapping. These methods are basically divided into two groups, whether they are meta-analysis based or joint analysis based. So I'm gonna next explain what are the differences of these two approaches. Actually, three. The first approach is the most straightforward, maybe, which is very similar to the GWAS meta-analysis approach. Suppose you have two cohorts. You basically run GWAS in each cohort, run a meta-analysis. You also compute a meta-analyzed LD matrix based on LD matrix from independent cohorts. Then you prime map this meta-analyzed GWAS and the meta-analyzed LD matrix, assuming this summary statistics actually came from a single cohort. And you might apply additional post-hoc QC to further remove spurious and false positive signals. Second approach is kind of similar to the meta-analysis approach, but this is particularly we call a post-hoc meta approach. Instead of doing a meta-analysis on the GWAS, we do a kind of a meta-analysis on the fine mapping results level. So we basically run a fine mapping in each individual cohort. Then you're gonna combine these fine mapping results, either taking minimum of the PAPs or maximum of PAPs across the cohorts. And the third approach is a joint modeling approach. So basically, the previous two approaches are analyzed analyze each cohort separately. But in this approach, you model two cohorts jointly. Presumably on a single server, you basically get the GWAS summary statistics and the LD matrix from each cohort, and you run joint file mapping on these summary statistics and the matrices. All of these methods works to some extent, but there are also challenges underlying on these methods. In particular, if you have multiple cohorts, you should always expect there's some heterogeneity across the cohorts. It could be phenotyping, like UK Biobank is a population-based, Biobank Japan is a hospital-based. So for disease phenotype, we observe a higher heritability in BBJ. But for biomarkers, you observe a higher heritability in the UK Biobank because biomarkers in the BBJ to some extent is influenced by these hospital-based recruitment. It could also be a genotyping and imputation. Each cohort, by nature, uses a different genotyping array and a different imputation methods. And these differences in phenotyping, genotyping, imputation inevitably creates a heterogeneity across cohorts that poses a substantial challenge in fine mapping. There's also issue of the lack of the in-sample LD or adequate external LD difference to conduct a fine mapping, but which is basically a separate issue. And again, the heterogeneity issue is a DR issue. This is one example from the COVID-19 host genetics initiative, where we basically observe in a well-known TIC2 locus, non-missense variant is not genome-wide significant, just purely because some of the cohorts are not well imputing this missense variant. So just because of the missingness, not because of the real biology, we miss this missense variant from the statistical fine mapping. And this poses a significant challenge in the underlying data, not the method itself, for conducting a multi-cohort fine mapping. To evaluate to what extent these heterogeneity influence fine mapping across the cohorts, we did extensive simulations. For example, we simulated different genotyping array under a different scenario and observe, to what extent that different genotyping arrays contribute to the host discovery rate. We simulated the different imputation panels that actually substantially contributes to the host discovery rate because of the differential missingness patterns. We simulated the genetic ancestry, which to some extent contributes to the FDR, but more importantly, having African ancestry contribute a lot to the improved recall or improved power. And this is because African population tend to have a shorter RD lengths compared to the other population. And finally, a mixed scenario where we observe in the worst case scenario, there's a host discovery rate of up to the 30 to 40%. And this basically demonstrates all of the meta-analysis fine mapping is significantly miscalibrated in the simulations. So to mitigate this phenomenon, we developed a method called slalom. The idea is very simple. Suppose you have a GWAS summary statistics like this. You can say there's a one causal variant that has 5 timing variants in a very high linkage disequilibrium, but one variant seems off. So we try to detect locus that contains this kind of outlier variants and identify loci that contains such outlier variants as a suspicious loci for fine mapping based on local LD structure. In the simulations, we show that although not perfect, host discovery rate in non-suspicious loci is 60% lower compared to the suspicious loci. And in the real data using the GWAS catalog, here we use the non-synonymous coding variants as a true causal variants because we don't know what is a true causal variants. We do see a substantial enrichment of the non-synonymous coding variants in the non-suspicious loci compared to the suspicious loci. Both suggesting several methods mitigate the issue of these heterogeneity in the meta-analysis fine mapping. So basically, our studies show that naive single causal variant or multiple causal variant fine mapping methods are often miscalibrated, and post-hoc QC, like a slalom, is very important. For a single causal variant assumption, we developed a method called slalom, and there's a recent paper that also models the multiple causal variants if you have a very good matching LD difference. And this method is called Calmer. And as you can see here in the example figure, where basically you have two causal variants, well, where you have variants, many variants in the high LD. In the Calmer, they correctly assign low but nearly equal PIP to the multiple variants. Whereas if you naively use a SUJI with the external reference panel, then you're gonna get many, like more than or nearly equal to 10 causal variants with PIP equal 1. So this Karma method works well in a situation when you have a good tau difference panel, like if you are doing a very homogeneous European meta-analysis, and this might be a additional support in addition to our proposed Saron method. You can also do a post-hoc meta approach. This is purely based on the assumption that if your single cohort file mapping method is well calibrated, post-hoc meta is often calibrated but less powerful. So suppose you have a UK biobank, Finjen biobank, Japan. In each biobank, you did a decent job of file mapping these geo-summary statistics with sushi. Instead of trying to do a meta-analysis or trying to do a joint modeling across the cohorts that gives you a extra computational cost, you can simply take a file mapping method from each cohort and combine results by either taking the maximum for most discovery, or either taking a minimum of a PIP for very consumptive method. And in this way, you can basically preserve the well calibration, but less power or less recall without doing additional joint file mapping or joint meta-analysis across the cohorts. All right. Then I would like to talk about the final category of the file mapping, which is a joint analysis. I don't particularly see a single causal variant multiple cohort joint analysis method. I'm pretty sure there is, but I don't think there's nothing widely or nothing outstanding. But in the multiple causal variant, multiple cohort setting, there's a plenty method development going on. One of the first multiple causal variant, multiple cohort method, file mapping method is called Painter, which was published in 2014, so 10 years ago, more than 10 years ago. This method is pretty, innovative at the moment, but unfortunately not very computationally scalable. Like you cannot, model multiple causal variants like more than 2 or 3 based on the complexity of depending on the complexity in your locus. Thanks to the development of the Suji, there is waves of the Suji extensions to our multiple cohorts. Namely, there are 3 methods proposed so far. First thing is ME Suji, Gao et al., Nature Genetics, Suji X by Yuan et al., also Nature Genetics. And there finally a multi-Suji method, Rosson et al., which is currently on the Met archive. These methods, by large, shares their same intention or same desire. You want to extend your Suji method into multiple populations, but there is methodological differences among the 3 methods. First of all, among the 3 methods, only the ME Suji only the ME Suji models ancestry specific causal variants. Suji X and the multi Suji, for the sake of the computational efficiency, assumes their causal variants are shared across the populations if they exist. I mean, if they are frequent in a given population. Based on their literatures, I think this shared causal variant assumption is very valid, but flexibility in the model gives liberty for ME Suji to model ancestry specific causal effect. Then for your effect size modeling, multi-Suji and ME Suji models effect sizes are correlated across ancestries to some parameter rho. This is based on observations or cross-population genetic correlation. And finally, on the post-hoc processing level, there is the Suji has a filter called PUT filter, which basically removes queryable sets that contains variants with R-square less than a certain threshold, so that if the queryable set for a single effect, if the variants are not correlated to each other for a given queryable set, then that queryable set is deemed to be non-pure and removed from the analysis, removed from the output. Multi-Suji paper basically finds that if you have this filter, maybe your queryable sets are pure, but their deco is substantially decreased. And Suji X basically implemented this PUT filter so that in the multi-Suji papers comparison, they actually compared Suji X with filter and the Suji X without PUT filter. But these are basically main differences in terms of the theoretical modeling. And in terms of the performance, these are performance comparison based on the simulations. On the panel A, we basically compare calibration. So what you can see here is, for PAP more than 0.9, a threshold, basically all the methods, all the Suji extension methods are very well calibrated, followed by PIP meta methods, which is basically taking the maximum PIP across the cohorts, and the naive meta-analysis based methods like NCB meta or SCB meta is not well calibrated compared to the other methods. In terms of the recall, again, three methods have a very comparable recall. Multi-Suji is the top, followed by Suji X and ME Suji, then PAP meta, and again, naive methods won't work well. Precision recall curve basically gives you the same conclusion, and panel D is important practically because if you run fine mapping methods, your computational cost is a huge constraint. And basically, multi-Suji authors shows that ME Suji is a prohibitory slow and they're memory intensive compared to the Suji X and the multi-Suji. So based on these results, we can conclude that these Suji extensions to the multiple cohorts methods have a higher potential and higher potential for fine mapping a cohort jointly at a Biobank scale. But this always comes with a caution, which I have to stress. These methods still suffer from the inter-cohort heterogeneity, especially missingness due to the imputation quality. multi-SUJI paper mitigates this issue by using all-BAS, the whole genome sequencing data. So all the cohorts they find mapped in their paper is a whole genome sequenced and from a single biobank. Meaning, we expect lesser heterogeneity across their populations. But if you want to find map, say, UK Biobank and MVP, then these two biobanks have a completely different phenotyping scheme, genotyping and imputation technologies, and you should really put extra caution when analyzing and interpreting these data. Using their these joint file mapping methods.
May I ask a question, Masa?
Sure.
In the last 3 minutes or so before the official Q&A, is there a way that you can sort of flag these regions where you have this heterogeneity and missingness so that you at least know not to look under those rocks as much?
Sure. So that's basically the methods here, slalom, which flags these outlier variants in a single causal variant, or Gadamer if you assume a multiple causal variant. These are the methods that mean to flag these loci and remove them.
And that can work with the multiple cohorts together. Thanks.
Yes. All right. So instead of summary, I would like to show this tentative best practice flowchart, which is beautifully designed by Lan. Suppose you have a raw data, and you want to basically find map your GWAS summary statistics. If you follow this flowchart, you need to choose what we believe as a best practice so far for file mapping. For our first part, if you have an individual level data, then you first have to do QC and the GWAS based on the standard procedure very carefully. But if your underlying individual data contains multiple ancestries, then we're going to recommend using multi-SUJI or SUJI-X for joint file mapping, or a PAP method for single cohort file mapping, then taking a maximum PAP across the populations. If your cohort is relatively heterogeneous, but still within a similar ancestry vein, I didn't cover this topic in today's talk, but there is a method to compute a covariate adjusted Tauri matrix, which is important if your cohort is heterogeneous. Then you can run a standard file mapping like SUJIN file map. And if you are homogeneous, computing sample ID and run SUJIN file map. So that's our recommendation if you have our individual level data. If you have our summary level data, knowing sample ID, then the meta-analysis, file mapping becomes more challenging, and you need extra caution. Your summary statistics is still from one cohort, or if your meta-analysis in your meta-analysis, all studies are from the same underlying cohort, like all of us, or whatever, as a single technology or single cohort, then you can probably carefully choose the external rarity difference that represents your summary statistics. Then you can Gadamer for multiple causal variant file mapping, or if you don't find a good reference ID, you can either run a slalom or ABF simply. If you have a meta-analysis from different ancestries, only thing you can do is a slalom because Gadamer is not currently handle that particular situation. So hopefully this best practice flowchart will guide you to some extent how you're going to do a file mapping in your future studies. And with that, I'd like to acknowledge my PhD mentors, Mark and Hilary, Jacob and Lang for their great support during our file mapping work, and the rest of the colleagues for their input for our project.